So this segment is on photosynthesis, which is the process by which plants store energy that comes from light in a chemical form, let's say glucose. In the process, it's going to take light energy and you combine carbon dioxide and water to form glucose plus oxygen. It's a little bit more complicated than that, but we're going to skip that. And those are the parts you need to remember. We'll cover these topics as we go through, and here we go. So the reactions that convert light, water, and carbon dioxide into sugars are generally broken up into two sets of reactions, but here is the general equation here on top, and this is a good thing to remember. So carbon dioxide plus water gives us dioxygen and glucose. It can actually be any number of smaller carbon byproducts, or the glucoses can be linked up with other sugars to make more complicated things, but this is a nice succinct way to think about the overall reaction, so stick with this. Again, there's two parts: the light reactions, and that's the top half of this cartoon here, as you go through this, make sure that you're really remembering what are the inputs and outputs of each of the different reactions. So this is an error. Carbon dioxide is not involved in the light reactions. All the light reactions are going to do is bust up the water and generate a highly reduced coenzyme here, NADPH, and energy to use in the dark cycles in the form of ATP. So really the light reactions give us molecular oxygen, energy in the form of ATP, and a reduced agent that doesn't want to stay reduced that we're going to consume in the Calvin cycle. So this is a good time to point out that the net effect of photosynthesis is to reduce the molecules. That's to get them to a higher energy state so that when we burn them through respiration, we're going to be releasing energy then. The same energy that we're pumping in here to store it. So again, water goes into the light-dependent reactions, as does light, and at the output you get a reducing agent, a byproduct, oxygen 2, and energy. The light-independent reactions, otherwise known as the Calvin cycle, take carbon dioxide and attach it to rubisco, which we're not going to get into in lab, and ultimately transfer those carbons through G3P and other intermediates into a glucose molecule. And so in the process you need to spend energy to attach those carbons, so we're going to consume the ATP, and you need to be adding those hydrogens to this carbon dioxide molecule to get it back to a general carbohydrate chemistry, which is 1 carbon for every 2 hydrogens for every 1 oxygen. So you see this is highly oxygenated. We need to add hydrogens. That actually comes from NADPH. One of the reasons we go into such detail in this is because a very similar set of reactions is going to be working in reverse when we get to fermentation in the next section. Okay, so again, when you're studying for this, remember to know all your inputs and outputs and the net goal of each of the different reactions. So the overall, the light reactions, which is to give us O2 and these intermediaries, and then the independent reactions to take carbon and get it hydrogenated, consuming energy and the reducing agent and giving us sugar. Okay, so the way that photosynthesis is engineered in nature is it uses a set of pigments to absorb the light and transfer those activated electrons to other molecules. And so it does this using light from the visible spectrum. You should remember Roy G. Biv from your general science education up to this point, representing red, orange, yellow, green, blue, indigo, violet. As you go into increasing levels of energy, that most energetic light up at the narrower part of the spectrum, here in the violets and blues, is what is preferentially used by the plants. And so chlorophylls, A and B, absorb in those wavelengths, and they're the principal photosynthetic pigments. But right on top of them, you also have carotenoids taking care of the rest of the blue spectrum. You'll notice there's a gully here before you get to some secondary absorbances in the red from the chlorophylls. And actually there's another set of pigments in here, xanthophylls, that actually absorb in these yellow and orange wavelengths. And so that part of the spectrum can be used by plants too. However, you'll notice there's nothing that covers the green spectrum, and so that's why plants why plants end up looking green to your eye, because the plant does not absorb any of those green wavelengths. In fact, it rejects them, kicks them back out of the tissue, and therefore they're there for your eye to detect green. I should point out that there's another set of pigments that are not photosynthetic. They are called anthocyanins, and their appearance is based on the pH conditions within the cell. A famous example of this is the hydrangeas that you see. Those big balls of compound flowers, and they're very stereotypically red, white, and blue. And so that's all dependent on the soil chemistry for each of those. So in this lab, there's plenty of reasons you would want to detect the rate of photosynthesis. You can see, like, if you're in agriculture, whether or not your plants are doing, they're photosynthesizing well, when will the harvest be ready, and so forth, those kind of measurements. And there's a variety of ways to detect them indirectly. And so you should be aware of these and aware of which one we actually use in class. So what you can do is you can measure the amount of carbon dioxide consumed. Remember, the net goal is to turn carbon dioxide into sugar. You can measure the amount of oxygen produced. Remember, that's a byproduct. Or you can measure downstream of the production of the glucose the amount of starch that is stored once those glucoses are polymerized. In our lab, we're going to measure the amount of carbon dioxide consumed. And for that, there's an important bit of chemistry with carbon dioxide in water that we need to address. So when carbon dioxide is dissolved in water, it forms this very unstable compound called carbonate. And you recognize this from your soda pop. If you shake it up, it busts up, and what results is the carbon dioxide leaves the solution of your soda pop, and the water is left behind, leaving a lower pH, we'll get to that in a second, and extra water, so it tastes flat. So what about this pH thing? When this compound is formed by dissolving carbon dioxide in water, this is actually a weak acid. So adding this compound makes the solution more acidic. You may remember we talked about in class the actual pH of water sitting out is actually closer to 5 than the 7 that they teach you, because it absorbs atmospheric carbon dioxide. Whereas, if you distill the water and make it nice and pure, that's also not the case. But anyway, when this is in solution, being a weak acid, it can dissociate to form the hydronium ions as well as this bicarbonate ion. And so this results in a lower pH when this substance is in solution. What we're actually going to do, just to confuse things, is to add sodium bicarbonate. So we're going to add sodium on this side and the same bicarbonate molecule, which without the hydronium molecules actually makes it a weak base. So it'll actually raise the pH to basic conditions. But the point is the same, is that we're going to be dissolving carbon dioxide in this solution to make it more accessible to the plants. Moreover, we're going to measure the amount of carbon dioxide that goes away. So we can measure that again indirectly by measuring the pH of the solution as it changes as the elodia photosynthesize in the same solution. While a bit verbose, I hope that does the trick. And what we hope to see is that anything that we give sufficient plants and carbon dioxide to that the carbon dioxide is consumed, and that results in a change in pH.